our model, we looked at what kind of things happen following this brain death, this herniation, the catecholamine storm. And you see here that there's a lot of very important inflammatory markers which are upregulated, and there's a lot of gene expression, which then will also lead to protein formation, and those proteins can be dangerous for the organs. So there's not just a cytokine storm, because cytokines come up and they go, so maybe in 5 or 10 minutes they will be gone, and you might say, well, does it really matter? A little bit of cytokines left and right? Well, but what you see here is they really start to go into the parenchyma, trying to find the pointer, where you actually have here adhesion molecules in the liver. There's PMNs, neutrophils who go into the parenchyma. There should be only 3 or 4 in a normal individual, or a living donor, for example, as a control. And you see a lot of these toxic neutrophils going into the parenchyma, causing a lot of injury. So they're the bad guys, for example, like reactive oxygen species. We know that the body doesn't like them because they cause harm to cells. But the interesting thing is, at the same time, there's also the good guys. So while we're having this very detrimental inflammatory response, we also have somehow some cytoprotection. So the system, despite the fact that there's unphysiological state of brain death and basically the individual is dying, there's still this kind of search for nature, for cytoprotection, to let the organs and the tissues survive, for example, by activating heat shock proteins or hemoxygenase 1, which is HO1. You could say, well, again, how interesting this is. Does it really affect my organ? Well, it does, because function deteriorates over time. And the more unstable your donor is, of course, the higher the inflammasome and the inferior your function. You can see here control versus marginal and optimal managed donors. There is a significant difference in function at the end of the donor period. We also did some models where we took brain death donor kidneys from the rats and then transplanted them into healthy rats and compared that to those who were not from brain dead, but more like, let's say, living donor rats, healthy rats. Significant difference, earlier rejection and lower graft survival and inferior function if you were a kidney coming from a brain dead donor rat, which of course we also know in human situations. Same for the liver. Here you see 3 groups. There's like the group A, which is the control, where you just did like 1 hour preservation and then transplantation of the liver. Then there's a short period of cold storage with some brain damage, and then already you see a reduction in survival. And if you combine now something with, as such, should not be a problem, the brain death plus a little bit more preservation, you see actually that there's a significant reduction in graft survival. So let's go back to the assessment. How well actually do we know what's going on in the organ? How well are our current clinical measures reflecting organ quality? So we have the donor medical and social history, for whatever it's worth. It's an indicator, we know. Is it really very helpful? Well, sometimes it is, sometimes not. We have virology. We, of course, for kidneys, do a lot of matching, and that could be helpful. We have laboratory data since admission, which somehow gives you a trend where we're going. There's radiology findings, but how well is creatinine, for example, predicting outcome after transplantation, whether a kidney is going to have immediate function or not? And then, of course, there's the magical surgical eye. As surgeons, we always believe in our own observation, and we always say, well, this is a very fatty liver, so we should not use it. And this is actually a very good liver, so let's transplant it. Well, there's several studies being done, and the magical eye is not as magic as all, because many times we think it's fatty and it's not, and the other way around. There's only about 20% of the cases the surgical eye is correct when you compare to histopathology. So I think there's something to win here, because there's a lot of uncertainty, and because of this uncertainty, a lot of organs nowadays are not procured, and they're not even transplanted, right? So what we have now is let's say a footprint. We have an indication of what's probably good, but as you can see, sometimes the sea can wash the footprint away within so many seconds, and then we have no information at all. So what we did actually in the UK is we said, well, we need better assessment of those qualities of donor organs. So we set up a quite unique national biobank, collecting samples from all the donors in the UK, and then allowing us to do research with different groups on the quality of the liver, the kidney, the heart, the lung, the pancreas, and so forth. And how do those markers affect outcome? Because what we think is we need not a footprint, we need a fingerprint, which is much more refined. And of course, the question is, what is a bad or a good outcome, and can we predict bad or good outcome, and therefore maybe match those organs with other recipients instead of with the wrong ones? Now, I'll leave it to you to say what is a good or a bad decision. I know what my choice would have been, but that's political, so I'm not going to go into detail here. So what we did actually then is we said, with our national biobank, we have the transplant registry, which in the UK is extremely good, with all the donor data and the transplant data. We have, of course, the donor management data, and then we add the national bioresource with all those samples, which basically are collected by our specialist nurses and retrieval teams while they're on the job. So it's not like an extra effort. It's an effort, okay, but it's like a minimal effort because it's been arranged quite well. We started some years ago, only 2 years ago. We have now more than 2,000 donors included in Quad, which is about 80% of the country, and we have something like 200,000 biobank items, including blood samples, urine samples, kidney biopsies, liver biopsies. We're now starting to collect lung and heart biopsies as well, bile ducts, because liver transplant surgeons are very much interested in the bile duct and the ischemic, of course, biliary lesions post-transplant. And of course, a lot of groups now, about 20 projects, are focusing on how can we improve the quality. This is just an example of what we did in our own lab, trying to see whether we could use proteomics, looking at the protein production, whether that would be yes or not affecting the outcome after transplantation. And we were actually able to already demonstrate that depending on the type of proteins, you were able to detect whether this was a DBD, or a DCD, or a living donor, because they all had different profiles. And you could also predict which one was going to develop delayed graft function, which I think is a start. But we are going beyond that, and I would like to share that with you. So we say, for example, okay, so at 1 year, we define together with our nephrologist colleagues what is a really good kidney. And I think you all would agree, if there's immediate function and a GFR at 1 year of more than 60 mils per minute, that is a very good kidney. It's very close to a living donor kidney. Now, if there's DGF in the beginning and a GFR less than 30, that is not a good kidney. And the chances that that kidney is going to fail in the next year is quite big. It's about 75%. So then we said, okay, so give us the selection of those kidneys, and then we go back to the kidney biopsy and the samples at time of donation, and are we able to find markers which tell us which kidney will develop good function or bad function? And you can actually do that by doing proteomics, and then using a very interesting kind of ingenuity pathway, you can downsize from those 3,000 proteins to about 10 to 15, which are really clinical relevant. And of course, the next step is now that we have to validate those. And if we have validated them, then we can maybe produce a kit which will help us during donor management to see what we want to see as far as predictability. So these are some of these markers, for example. This is an activation of apoptotic pathways, where we have this suboptimal group and the good group. We also have a living donor group as a, let's say, gold control, because the living donors obviously are wonderful. They have a very good function. There's also some other already very early indicators of kidney fibrosis. Let's not forget that most of the kidneys nowadays are lost due to chronic allograft dysfunction, let's say 10 years later. And many of these patients come back to our waiting list. It's about 30% of our waiting list now. So this fibrosis is very important. But there's also cytoprotective pathways. And of course, it's very interesting now to find out how can we define the balance between the good response and the bad response. So we will hopefully be able in the next year to say which kidney goes what kind of road up. So here you see all the influences you can have, which is either in the suboptimal or in the good kidney function, and it's about the balance which is, of course, important. Why is this relevant? Well, one of the things is, if brain death is so hostile, shouldn't we just run and take out these organs as quick as possible and not let them sit very long in this very hostile environment? So we said, well, what is actually the impact of the duration of brain death on the outcomes? Because if it's really bad, we should probably rush and retrieve. So one of our DPhil students, Kat Buffer, did a very interesting study using this transplant registry data in the UK, which I think is very good and granular, and looked actually at the effect of the duration of brain death on outcome. Our current brain death duration before retrieval is about 30 hours, and in a DCD donor it's about 21 hours, which is very long. So there was a lot of material which we could study, and the interesting thing is that there's actually a positive effect on prolonged duration of brain death. So it is probably less hostile than we think, because if you have the possibility during those, let's say, 20 or more hours to optimize donor management in your intensive care, the outcome of those kidneys is much better. For the pancreas and the liver, it was very difficult to see in those numbers, but also for the heart and lung, again, the same was seen, better heart transplant results after a longer duration of brain death. Well, that's a positive fact. I don't mean we always have to do that and let it be so long, but it means that we actually can improve something during that period. And there's a lot of natural factors we could actually use. I just picked out one which is appears to be quite important and where we already can do something, because during that period of brain death we can optimize, but we can also maybe add some additional agents which can improve the quality of the organs. Now, complement is significantly activated during brain death. And we have tested this in the animal model, but also in the human model. There's an enormous upregulation for plasma C3d, which is one of the steps in the complement chain, versus control. And we then added a soluble complement inhibitor, which is a very safe agent currently on the market, used for other purposes. And it was actually, as you can see here, we were able to reduce that activated complement to quite low. We gave it before and we gave it after. And of course, after is more interesting because you don't know who's going to become brain death. But if you can give it after brain death and it's reducing the complement activity, that could be beneficial. Then we took those kidneys from those animals which were inhibited and transplanted them in healthy recipients. And as you can see, better survival and quicker function restoration to normal in those that had been inhibited. So the complement inhibition basically benefits the outcome after transplantation. And that is something which we also could do in the human situation. And these are the agents which are already in the market. So they're already found to be safe, but we have to start to do such kind of studies. Finally then, a small part of the preservation, the rest will be in the other talk. An important factor in organ preservation is what? What is the most important factor in organ preservation as we speak? Temperature. Thank you very much. Great. Temperature effect. And what does the temperature do?
It keeps the cells in a sort of suspended state rather than active metabolic state.
Okay, reduced metabolic state. Sorry, you said?
Acidosis.
No, sorry. The ladies are talking now. Yeah. You can't do your emails and listen to me and to the ladies. That's impossible. So say it again, please.
I think it prevents any kind of autolysis.
Yes. Yeah. So degradation of proteins is reduced. So in a way, we wash out, we rapidly cool, we therefore reduce ischemic injury, we keep it suspended, and we think that therefore no nutrients and no oxygen is required. And it's like keeping your meat in the fridge, it just keeps it a little bit longer. But of course it doesn't improve. It will still kind of go downhill, but it will maintain quality a little bit better. What else is important in organ preservation?
Excuse me, sir. Can we see the previous slide, sir?
The previous slide?
This slide, yeah, this one.
Did you recognize one of your family members?
I was just noticing the temperature.
Yeah?
It's written 0 to 4 degrees. So is that the optimum
Well, the thing is this. I think what we ended up is there's a coefficient that with every so many degrees, there's a 10% reduction. And if you come down to about 5 degrees Celsius, your metabolism is about 90% reduced, which is sufficient for the cells to survive. They will not freeze or crystallize. And if you then restore ATP, they're able to function. You could go up to 10, but if you go higher than 10, then you have to add oxygen and you have to have breakdown products, so you have to somehow clean the system, otherwise there's degradation. So 5 is in a way found to be the best optimal cold temperature, unless you really go to freezing, but then you get other problems, right? So washout. We all think, and we heard that just during our beautiful demonstration on the donor, that washout is so important because you want to rapidly cool the organ, put the slush on there and the ice, because you want to go down from 37 degrees to 5, because then metabolism is safe and warm ischemia is bad. We all agree, right? Well, guess what is the temperature when you flush an organ and you have your 5 liters of UW infused? What do you think is then the liver temperature? So now,
Sorry, which temperature?
the liver temperature.
When you start flushing, it will be.
No, well, after 5 liters. So you're done with your preservation, and it's 5 liters down the drain. You spent like a lot of good money, but it's a beautiful solution, of course. And what is then the temperature of the liver?
The core temperature should come down, and the core temperature should be between.
I think 15 to 20.
You wanted to be at 5, right?
Less, 10.
10.
10.
Who be more or less? Come on.
15.
Mid 30s? Or mid 20s? Good.
After you flushed it, he stopped.
Yeah.
Yeah, okay. Well, this is a line for the liver in the kitchen. We had these, you know, in the food industry, they have these guns, these laser guns.
Yes.
So we did a lot of series of human donors where we just have our transplant coordinator look at the temperature. The interesting thing is, it doesn't come below 20, despite whatever you do. And actually, when you compare HDK and UW and Marshall's, actually UW is still the better one, probably because it contains a colloid, and as the colloid is a little bit larger, it stays better in the vasculature and isn't flushed out that quickly. HDK doesn't have that. So temperature is lukewarm. This is warm ischemia time. So if the cardiothoracic surgeon takes a little bit longer to take out the heart and the lung, our liver, pancreas and kidney are sitting there at 20 degrees for another hour. I'm not blaming the cardiothoracic surgeon, well, a little bit, but I think that we have to realize that is warm ischemia.
No, do you put then the slush ice while you're waiting?
Yeah.
Does that help to cool down it a little more?
Despite the slush, this is a test with cold saline and slush, and still getting below 17, 18 degrees is very difficult. Let's go to the normal cell function. Here's the normal cell. You remember school days. High potassium in the cell, sodium outside, mitochondria, our little energy factories making ATP, which drives the pumps. The cell has this so-called Donan equilibrium, which basically is a charge, making it impossible for sodium to enter, and it keeps potassium in, and that's why these pumps are so important, which are all ATP driven. Now, what happens during hypoxia and hypothermia, there's oxygen and there's cold temperature, so the pumps slow down. They don't stop, but they slow down. As a result, there's a lot of attraction of sodium, which is now coming into the cell because the cell is loaded with proteins. Proteins are negatively charged. That attracts the sodium, and with sodium comes water. So when you have hypoxic conditions and hypothermia, there's a lot of influx of sodium and water, which of course will mean, what do you get? Cell swelling, correct. And the result of cell swelling is? Yes, lysis. Good. So what are the, let's say, the 4 classic unwanted effects of hypothermia, which we all face, because that's the way how we preserve our organs, but we just have to take them and cope with them. So, one, we just said. Cellular edema. What's next? What else? Cold temperatures, low metabolism. What do you get when you have low metabolism with your ATP and ADP, AMP? Acidosis, right? What else? Other ideas? Yeah, very good. Did you listen to my talk previously? Okay. So there's the cell swelling, impaired energy metabolism, acidosis, and the precursors of the ROS, as you say, Sunil. So the choice of the components is also important. So we have temperature effect, but also solution effect. So you have to build a solution which somehow tries to kind of alleviate these things. So the next thing is then physical environment in a solution is very important. And what we can do actually to counteract is this. To reduce cell swelling, we have to provide buffering for the acidosis. And if we want to reduce cell swelling, there's some tricks. One of the tricks is that you add osmotic pressure. So if you flush the organ with large molecules like dextrans or starch or something really big, it will stay in the vasculature and basically try to prevent flux of water from the vasculature into the interstitium. In the interstitium, you then have saccharides or anions and electrolytes who can be very effective to prevent that this fluid, this water, is coming from interstitium into cell. And that's why the saccharides are so important. Glucose is the smallest, it's the least effective. Mannitol is more effective, sucrose even more, and raffinose is the biggest sugar. So there is a size and an effectiveness of those sugars. Negatively charged anions are also helpful, like lactobionate and gluconate. Citrate, like in Marshall's solution, the only disadvantage of citrate is it's a very small molecule, so it will enter the cell eventually. So oncotic pressure with Hess, starch, polythene glycol, quite big, and dextrans is very helpful, and this will effectively reduce cellular edema. There's another environment which could be important, and that's the so-called biochemical environment, which comes down to that these are agents we add to solutions because they may positively affect metabolism. So, for example, we all need ATP when we reperfuse. And since during hypothermia, the ATP is broken down within milliseconds, AMP and ADP and adenosine are just floating away. So if we provide adenine, ribose, and phosphate, which are the substrates for ATP, in the solution, it is there in abundance. So when there's reperfusion, the cell can easily take the components and make ATP. And that works. Antioxidants, very important. The only one which really has been shown to be effective is glutathione. The rest sounds good, but was only good in the mouse model, but not in human work. A lot of other factors which have been tried, many of them very good experiments, beautiful PhDs, unfortunately in the clinical work not really very effective. This is just to show you the glutathione, how effective it can be. These are rabbit renal tubules, but you could also do with the liver slices. You incubate them with or without glutathione, and then you look at the ATP production and at the LDH production. Now, LDH is a very aspecific general marker for cell injury. So high LDH is bad, because when the cell dies, it comes free. So that's why it's a nice marker. And what you see actually is if you add glutathione, there's better ATP production and lower LDH versus controls. And that is also something which we have tested in kidney transplantation and liver transplantation. The organ is just better surviving and functioning if the glutathione is added. So this kind of, all these kind of little tests and experiments led to a more systemic approach, and that's how Belzer and Southard in the 80s already, which is like ages ago, started to kind of develop new solutions, and one of those solutions was the University of Wisconsin solution, which until now is, for most centers and for most organs, still the gold standard. Some newer solutions have been developed, but many times they will be look-alike. And again, to kind of quickly summarize at the end of my talk, is that the effective components in those cold storage solutions is that there's a sodium-potassium ratio. There's a long discussion whether you should have high potassium or high sodium. In the end of the day, probably high sodium is as good as high potassium. High potassium has the disadvantage of having this kind of vasospasm during the first so many minutes. Otherwise, it's fine. There has to be a buffer and the impermeance, as I just said. The more, let's say, complex the solution, the better it works. So urocurrents, for example, or PBS are the simplest one, work not as well as the bigger ones. Special scavengers and the osmolality. Quickly, the trial, when we did the UW solution versus Eurocollins, we saw a significant reduction in delayed graft function between the two groups. Then HDK came to the market, which showed within normal cold ischemia times no major difference with UW solution, although we found out now that with prolonged periods of cold ischemia time and more difficult donors, older donors, especially in liver transplantation, there's more complications with HDK than with UW, which has been reported by different groups for both liver and kidney. New solutions were also lookalikes, Celsio and IGL-1. Basically, they're the same as UW. Remember, they have these electrolytes that they chose for the high sodium, fine. Then they had impermians. Celsio actually kept the lactobionate, added the mannitol. In IGL, it's identical to UW. Histidine instead of phosphate, fine. And they basically did not take a colorate into account. IGL added instead of the Hess, which is the starch, polyethylene glycol. Well, in clinical situations, the studies that have been done are very equal to UW solution. Unfortunately, most of those studies are a little bit underpowered, so it's never been a real trial. Why? Because you need thousands of transplants to prove non-inferiority. Finally, then, the last slide is, well, I've told you about cold storage, and we've been very happy during the happy days of organ donation when we had young, healthy donors. The question is whether that's the way to go with these older and higher risk ones, because originally when we started with organ transplantation, it was machine preservation. And I'll come back in that later talk after the Skype. So I'll keep that for now as an open question, because we have now a wealth of techniques and novel ideas how to work in preservation, but we still have no clue what to combine when, for which type of donor, and I'm going to tell you a little bit about that in my next talk. Thank you.